So if you recall the promoter structure video, the link is in the description if you want to check it out. We discussed a simplified structure of the gene, which is that it contains the promoter sequence that initiates the process of transcription. And then we have the transcription start site, which is the beginning of the coding region, and that's the DNA code to make the RNA. And this happens in a process called elongation. And now, once the code is read, we need to stop at the end of the coding region. And this happens in the process of termination, which is the focus of this video. So apart from just not letting the elongation continue, why is termination important? Well, the obvious answer is not so obvious until we see that in a genome, multiple genes are present in tandem. So if you don't want the downstream genes to be expressed, the termination must happen at the end of the defined coding region. Also, let's say you need to make more RNA of the same gene. And in that case, you might want to recycle the RNA polymerase so that it goes back to the promoter and starts over again to make more RNA. So before discussing the main mechanisms of the termination, let's discuss the problems or issues related to termination. So looking at the structure of the gene, we know that we need to terminate the process at the end of the coding region. And this needs to happen within a certain window, else the elongation complex will just keep moving on. So if we focus at the end of the coding region, we should note that the ideal stop would occur exactly at the end of the coding region. And you can't have the elongation complex terminate earlier in the coding region because you would lose information at the end of the coding region. But it is okay to stop after the coding region because in that case you wouldn't lose any information in the RNA. But stopping too far is not favorable because then it is a waste of energy. So our termination happens in a sweet spot at the end of the coding region, and this is usually called the termination branching point, sometimes also known as the kinetic window. Now, if we think about the steps that need to happen, we can imagine that the polymerase, which is in an elongation complex, needs to slow down and pause. After this, a conformational change must happen that prevents elongation complex activity, meaning that a paused elongation complex must not resume elongation. And then the RNA-DNA hybrid needs to dissociate so that the elongation complex can disassemble. So there are two main mechanisms that are involved in the termination process. The first one is called the intrinsic termination mechanism. This mechanism is largely driven by DNA and RNA sequences, which mainly involves the formation of termination hairpins, which are nothing but secondary structures that the transcribed RNA forms. And this causes elongation complex rearrangements. The second mechanism is called the extrinsic termination mechanism. It is called extrinsic because it requires the help of extra factors, such as proteins that assist in the termination process. Now commonly, this extra factor is the Rho protein. Sometimes the extra factor can be the MFT protein. But Rho factor is more commonly studied as the extrinsic mechanism. And sometimes, for that reason, we also call this mechanism a Rho-dependent termination mechanism. But if we were talking about MFT, we would call it an MFD-dependent termination mechanism. So we'll just simply restrict our extrinsic mechanism to Rho-dependent termination mechanism. So Rho is a ring-shaped protein, which is an RNA translocase that binds the RNA. So in simple form, we can imagine how this would look by looking at this cartoon. The Rho protein binds the transcribing RNA at specific locations, and it would translocate until it hits the elongation complex. The movement of Rho is of course from 5' to the 3' of the RNA. Just one quick note on the terminology. If the extrinsic mechanism is equivalent to saying Rho-dependent termination mechanism, the intrinsic termination mechanism therefore can be considered an equivalent of Rho-independent mechanism. Now we can discuss these two mechanisms in more detail. First, let's look at the intrinsic termination mechanism. And as we just said, this mechanism is primarily driven by the RNA and DNA sequences. Actually, it is the information in the DNA that gives rise to the RNA sequences that have the potential to form secondary structures in the transcribed RNA. To understand these RNA sequences, we first need to understand the termination sequence. The structure of the template DNA of the termination sites looks something like this, where you have some thymine followed by a GC-rich region, which is followed by a few adenines. Now this template is used and the RNA is transcribed. So you would have corresponding to the template a complementary sequence. Now in the RNA, this A-rich part is called the poly-A tract. And at the end, you have the poly-U tract, which is about 8 to 10 nucleotides in length. And in the middle, we have this GC-rich region, which is about 8 to 10 nucleotides in length. This GC-rich region is a bit special because the bases in this sequence can base pair within themselves. And these are primarily the G's and the C's. This base pairing in the GC-rich region gives rise to the structure known as the terminator hairpin. We can draw out this hairpin better. Here we have the poly-A tract at the 5' end of the RNA, followed by the GC-rich region, which is self-pairing, followed by the poly-U tract. This entire structure is called the hairpin, and the GC-rich base-paired part in the middle is called the GC dyad. Let's take a step-by-step look at the process of intrinsic termination. The first step of the mechanism is that the elongation complex needs to begin pausing or slow down. This is important because the elongation complex is traveling on the coding region at a speed of about 60 nucleotides per second. And to terminate within this short kinetic window, it needs to slow down. The pausing is initiated by the poly-U tract as soon as it is transcribed at the transcription bubble. Let's quickly draw out the elongation complex in this process of slowing down. So in this state, you have the poly-A tract hanging outside, the GC-rich region is in the RNA exit channel, and this GC-rich region is not present as a hairpin structure yet. And in this active site, you have the transcription bubble, where we have the poly-U tract. And this causes the elongation complex to slow down. I will use EC from here on to refer to elongation complex. And this slowdown leads to the second step of the process, which is the formation of the transcription hairpin. This initial stage of hairpin formation leads to the formation of a weak hairpin. And we will see why that is in the moment. Now notice in the previous step, the GC-rich region is in the RNA exit channel, and the slowing down of the EC provides time for hairpin formation. And now if you look at the elongation complex in this second state, we see that the U tract is still in there, but the GC-rich region forms a weak hairpin structure in the RNA exit channel. And this actually causes the slowed down polymerase to pause for a moment. So why do we refer to this hairpin in the RNA's exit channel as a weak hairpin? Well, if we look at the structure of this hairpin, we see that in this hairpin, we have a poly-A at the 5' end, the poly-U bound to the template DNA at the 3' end, but the GC region actually has some unpaired bases that can still base pair. Therefore, it is referred to as an incomplete or weak hairpin. And the transition from weak to strong hairpin is largely a case of favorable energetics, which leads to the third step of the process, which is the complete hairpin formation. And once this hairpin formation is complete, the elongation complex is deemed committed for termination process. And then it is also no longer considered in the paused state. And this also suggests that the complete hairpin formation makes the termination process irreversible from this point on. Just to draw out the complete hairpin for clarity, you have all the G and C's paired. Now this also leads to some conformational changes in the elongation complex. A major one is the folding of the trigger loop, which blocks the E site. Normally, trigger loop is in an unfolded state. If you want to learn more about the trigger loop, you can check out the transcription elongation video when we discussed the RNA polymerase structure. So after the elongation complex commits to termination, it moves on to the final step, which is the disassembly process. There are two main models that have been proposed for disassembly, and these are called hybrid-shearing and hyper-translocation. The hyper-translocation is fairly straightforward. In this, the elongation complex just moves ahead without any addition of the NTP, and the trigger loop, which is blocking the E site, helps in this process. So if we look at the complex, we see that the transcription bubble simply translocates while the E site is being blocked. And this causes the disassembly of the complex. In contrast, in hybrid-shearing, it is the rotational force of the hairpin that is utilized to cause the unwinding of the RNA and DNA at the transcription bubble. So just a visual representation of the complex is that the force comes from the rotation of the hairpin, which is sitting in the RNA exit channel. So why does the unwinding cause disassembly of the elongation complex? If we look at the transcription bubble in this complex, we see that it is largely a U-rich sequence. And if you break all these bonds in the RNA-DNA hybrid, it will try to reanneal, but because it is repetitive, it will often reanneal incorrectly. And in this example, the TCA cannot be accommodated. And this destabilizes the RNA-DNA hybrid. Now, because UA bonds are easier to break than GC, it is thought that the hybrid shearing often occurs in a U-rich transcription bubble, versus the hyper-translocation is favored when there are less U's. There is another model that is also used to explain the process of elongation complex disassembly, and this is called the hairpin invasion model. In this process, the 3' end of the RNA actually stays at the I+1 and the I site. Again, if you want to learn more about this, check out the elongation video. So the RNA does not move at the 3' end, but the poly-A tract, which is at the 5' side, causes an allosteric inhibition of the complex. To see how this works, you have the RNA with its poly-U site at the transcription bubble, which is paired to the adenines in the DNA. Now the poly-A from the RNA moves into or invades the active site, and this breaks the RNA-DNA bonds. This results in a much larger hairpin structure, which is also now sitting in the active site. So you have this GC loop, but now on top of it, you also have the poly-U's and the poly-A's bound to each other. And the template DNA is now single-stranded. So this larger RNA hairpin is unfavorable for the enzyme, and this causes the elongation complex to disassemble. And that is the process of intrinsic termination. Now let's talk about the second mechanism, which is the extrinsic termination mechanism, also known as the Rho-dependent termination mechanism. So Rho protein, as we said before, is an RNA translocase, which is a member of the Rec-A family helicase. It is a hexameric protein, meaning that it is made up of 6 monomers. The hex here means 6. So let's see the simplified structure of the monomer. It has the N-terminal domain and a C-terminal domain. And when you get 6 of these monomers together, you form a hexamer. And that looks like a ring structure, where the N-terminal domains are positioned outwards and the C-terminal domain is in the inside. The empty space in the center is called the central pore. The N-terminal region has the binding capacity for C-rich and G-poor RNA. And this is also the primary RNA binding site, meaning that the RNA binds here first. Then CTD is the secondary binding site. The C-terminal domain also harbors the ATPase domain, which is involved in the process of Rho translocation. Just to expand on these C-rich and G-poor primary binding sequence, these are also known as the Rho utilization sequences, sometimes for short referred to as RUT sites. And they're about 80 to 90 nucleotides in length. If we look at the position of RUT sites with respect to termination and elongation complex, we find that the RUT sites are actually located upstream of the kinetic window or the termination site. Okay, so the first step in the process of extrinsic termination mechanism is the recognition of RUT sites by the Rho protein. Again, if we visualize the elongation complex, we notice that the recognition happens when the enzyme has actually transcribed the RUT sites. And these are, as we said, about 18 nucleotides in length. So these sequences are present in the RNA. Looking at the sequence structure of these RUT sites, we don't find a strong consensus except there is this periodic YC dinucleotide enrichment, spanned with some random sequences. The Y is an IUPAC symbol, which means that it can either be a C or a T. So we can say that these dinucleotides are pyrimidines. Now these dinucleotides are recognized by the N-terminal domain of the Rho monomers, and we have 6 of them. If you want to draw out this recognition step, you would see that the Rho positions itself with the transcribed RNA, and the dinucleotides bind at the N-terminal domains. And the random nucleotides between these dinucleotides just dangle around. The 3' end of the RNA, of course, is part of the elongation complex, which is still elongating on the DNA, while the Rho recognizes the RUT sequences. Something very special about the N-terminal domain is the presence of specific amino acid residues, like Q-loops or cold shock domain units, or some helix bundles that are involved in this dinucleotide-specific binding. The Q-loop in particular is very important, because it likes to bind these pyrimidines. And it also looks for 2' hydroxyl of the RNA sugar, or specificity. Now, notice that the DNA is 2' deoxy, so it doesn't have the same sugar. Therefore, the Q loop is very much responsible for the RNA-specific interactions of the Rho protein. And this bound state of the Rho protein is called the open ring state. And this binding causes the isomerization of the Rho protein, which leads to the formation of a closed ring state. So in the closed ring state, the RNA moves to the inside of the Rho protein, which is the central core, and then this initiates the secondary binding of the RNA by the C-terminal domain. These bindings involve 5 base pair of RNA, and that's because that's the approximate length of the central pore. So now if we draw out the closed ring state, we have the same structure at the N-terminal domain, but the RNA now is looped or threaded via the pore. And just as before, we have the 3' end of the RNA still with the elongation complex. In the C-terminal domain, you likewise have different units, like Q loops or R loops or Walker motifs that help in the RNA binding. And this, unlike the N-terminal binding, which is specific to the YC dinucleotide, the C-terminal domain does not care about the RNA, so its binding are very nonspecific. So now, once this closed ring state is formed, this activates the ATPase domain, and the Rho complex then moves on to the second stage, which is the translocation stage. In this step, the Rho uses energy from ATP catalysis to move along the RNA. To draw out the simple cartoon of this, we have the ring-shaped Rho and the RNA, which is bound to the Rho, and then you have the elongation complex on the 3' end. The Rho now uses ATP to move. Important thing to keep in mind is that this movement is 1 base pair at a time, because CTD is making nonspecific contact in the central core, so all the RNA-protein interactions have to be reestablished with each translocation step. This is very similar to the movement of RNA polymerase, which we discussed in the elongation process. And this style of translocation by Rho is called the asymmetric movement. One super important thing to keep in mind is that the rut bindings are not broken when the Rho translocates, so they remain tethered all the time. So how does the translocation look like? When the central pore is translocating 1 base at a time using ATP, the RNA is actually looping out while the rut sites remain bound. And if you think about this, now you get 2 complexes, the Rho and the elongation complex, moving in the same direction. And therefore, there's a translocation competition going on. And this competition is known as the kinetic coupling. And when the Rho proteins wins this competition, you get to the final step of the termination process, which is the elongation complex disassembly. And then we have the same 2 models to explain the stage as well. The first is the hyper-translocation, which in this case is also called the push mechanism, meaning that when the Rho protein approaches the elongation complex, it exerts a force onto the elongation complex, which reduces the time that the enzyme has to add any NTPs. Therefore, the enzyme just moves, and this destabilizes the transcription bubble, which leads to termination. The second model is the hybrid-shearing, also known as the pull mechanism. In this, the Rho protein approaches the elongation complex, but the dominant force now is the looping of RNA. So the Rho pulls out the RNA from the RNA-DNA hybrid by breaking the bonds, and this leads to the elongation complex disassembly, which terminates the transcription. Before we end this video, I wanted to leave you with some interesting facts about the Rho-dependent termination. First, that it is highly specific to the messenger RNA. In case of tRNAs, they're too short for the Rho protein to act, and in addition they have many secondary structures that would prevent Rho translocation. The rRNA, on the other hand, also has a lot of secondary structures, and on top of it, ribosomal RNA has these anti-termination sequences known as NUT sites, which stands for N-utilization sites. So how do these work? If the Rho is translocating on the RNA, you can have some hairpin structures or NUT sites that bind some proteins like the protein N downstream of the Rho protein. So in these cases, the Rho will not be able to get passed through them, and therefore the translocation of elongation complex will continue, and the Rho will not be able to stop them. So now the question is, how does ribosomal and transfer RNAs terminate their transcription? I would encourage you to think more about this question, and if you have any ideas, leave your answers in the comment section.